Okay, good morning, everyone. My name is Sarah Kostick. I'm one of the seminar committee members for the Plant Breeding Center. This is the first seminar of the year. There's typically about one per month, so put on your calendars October 10th. We will have our next seminar. I feel like I don't have to introduce Rex, but I'm going to do it for formula's sake. This is Rex Bernardo. That's going to be giving our first seminar, which is going to be a straight up chalk talk because we can't get the projector to work. He is a distinguished university teaching professor and an endowed chair in maize breeding and genetics, undergraduate from the Philippines, and then a Ph.D. from the University of Illinois. He invented genomic prediction. And most of his work is focused on marker-assisted selection in the past. He's also the director of the Plant Breeding Center and has written multiple textbooks. Most of us he's read multiple times, at least preparing for our prelims, and then he teaches multiple graduate and undergraduate courses. Rex, take it away.
Well, good morning. Thank you for coming to this seminar of the Plant Breeding Center. The theme for the seminar series is international plant breeding. And so the topic that I've chosen today has, I think, much meaning in international plant breeding. So I'll be talking about plant genetic resources from a breeding perspective. And I was also looking at alternative titles. A finalist was genetic diversity from a breeding perspective. And so you'll see that in a way I'm using genetic diversity and plant genetic resources a bit interchangeably. And a third title that I had in mind was my opinion of the most important topic that needs to be studied in plant breeding right now. And I think that really is the utilization, the effective utilization of plant genetic resources. Now, before we dive in too deeply, I think it's important for us to have a definition. What are plant genetic resources? And so to answer that question, I Googled it. Hey, plant genetic resources, how do you define that? And one definition that came up was plant genetic resources are the total hereditary material of a crop species and its relatives. Like, okay, I think that's a good definition. Plant genetic resources. You have a species you're working with, and you look at all the totality of the germplasm, the plant genetic resources at your disposal. It includes cytoplasm, it includes nuclear genes, it includes that species itself, it includes relatives that you can cross to that species, and that's then plant genetic resources. As I think about that definition, though, from a plant breeding perspective, that's more of an academic definition of plant genetic resources. And when I think of the breeders that I know, I'm in maize breeding, so a lot of my thinking is colored by my maize breeding experience, corn breeding experience. That might not be how they would define it. Because according to that definition, plant genetic resources would include the germplasm in the breeding program, it would include land races, it would include heirloom varieties, it would include germplasm stored in seed banks, and not just that species, but also relatives. And when I think of how a breeder might define plant genetic resources, I would define it as the germplasm that I don't normally use in my breeding program. That's plant genetic resources. And I see some nods over here. Yes, in a way, a breeder would say, okay, this is my breeding germplasm. That's what I work with. That's what I found to be productive. And whatever else that I don't regularly touch, that's plant genetic resources. It's kind of good to be there, but I'm not necessarily going to use it. And I had this pleasure of having a conversation with Professor Don Rasmussen. And some of you know Don, and he was a very successful barley breeding breeder, and Kevin Smith has taken over his program since then. And I remember this conversation in the hallway in Hayes Hall by the elevator, and Don told me, quote, I tell my opponents to use genetic diversity. That was how he viewed genetic diversity, and in a way, plant genetic resources. That he was perfectly happy with the germplasm what he was working with. It was very narrow, if you look at, based on Morex and Robusta and so forth, these historical barley varieties. And he says, People I don't like, my opponents, my enemies, I tell them, yes, use plant genetic diversity. And I think that that thinking can, in a way, this is might be my single slide, can be summarized in this picture over here, which says it takes more than genetic diversity to guarantee performance. And so here you have this guy on a sled, and some sled dogs with, I don't know what these dogs are. But you quickly realize that this sledding team is not going to win any prizes. It's not going to come first in any given race. And for many a breeder, this is the view of plant genetic diversity. And this is the view of plant genetic resources. Yeah, they're kind of good to have. They're kind of good to have in my pocket somewhere, but normally I'm not going to touch them. I'm not going to use them. If something happens in the future that I would need to dip into that, I might use that source of genetic diversity. And I think it's also reflected in the way that breeders work. If you think about how breeders work, they make a lot of crosses, they generate a lot of progeny, and they throw away most of them. They throw away most of them. As opposed to the way that things are viewed in plant genetic resources, instead of throwing them away, we keep them. We keep them, we keep seeds, we catalog them, we characterize them, because they could have some value in the future. And so I think that in a way, the view of plant genetic resources from a breeding perspective, it's a bit fuzzy. It's a bit nebulous. We would say, yes, we're all for plant genetic resources, but we're not going to really use them. And it's something good to have. And so my objective today is to stimulate some thinking and to make this idea or thought of plant genetic resources and plant breeding to be a bit less nebulous, to be a bit less fuzzy. What exactly are we talking about? What exactly are we dealing with? So I'll try to answer 4 questions this morning. Number 1 is, why would we want genetic diversity or plant genetic resources? Why? The second is, in what packages does plant genetic diversity come? Number 3 is, where do we find plant genetic diversity? And the 4th is, how? How do we use effectively plant genetic resources and genetic diversity in a way that's meaningful in the breeding program? So the why, in what packages, where do we find them, and how? That's the menu for this morning. Let's talk about why. And I think part of the why is we just look at history and some lessons that we've learned from history. For example, I think most of us are familiar with the potato blight that happened in Ireland in 1845 to 1852. And Ireland lost about a quarter to a third of its population, either from death or from emigration from that island. Why? Because the island, the farmers grew one particular variety, Irish lumpkin, that was susceptible to the blight. And the weather conditions were favorable for that disease. And the economic situation, political, social situation was that there were landowners, there were tenants working, and the tenants just had a small parcel of land on which to grow their food. And they found that really potato was the only crop that would meet their needs, because potato is so productive. It was the only crop that would meet their daily sustenance. So that's what they had to grow. And when the disease came, then it led to a lot of problems. And the famine ended only when more aid came and the weather conditions changed, so that the disease, the pathogen from the disease triangle, wasn't able to wreak havoc as it has before. And then more recently, in 1970, we well, I don't know if anyone, no one was around that time here. Or you were around. You were around. Okay, Les was around. Okay, Les can tell us all about the southern leaf corn blight epidemic in 1970, caused by the wide use of the Texas race T cytoplasm in hybrid seed production in corn, and that is a lesson. And so one reason that we would need plant genetic resources is for defensive purposes, so to speak, for defensive purposes of the plant. In case a new pathogen comes up, in case a new pest comes up, in case a new race comes up, in case of new weather patterns, hotter weather, drier weather, colder weather, whatever it is, for defensive purposes, it is good then to have plant genetic resources. And I think if you scan the literature, it has been said that some of the learnings, if you would, from that 1970 plant epidemic in corn and southern corn leaf blight is that we need to guard against genetic uniformity. That situation should not happen again. But guess what? Well, I think that's easy to say, but not easy to do. And I read this in a review article for an American Phytopathological Society. But take, for example, Dr. Norman Borlaug got his Nobel Peace Prize in 1970, the same year the epidemic, southern corn leaf blight epidemic was going on, 1970. He received the Nobel Peace Prize. Much of his work had to do with the introduction of dwarfing genes in wheat. Well, most of the wheat today, I don't know if there's still any wheat that does not have these dwarfing genes. So in a way, they're genetically uniform for these dwarfing genes. What if there's a pathogen, a problem we don't know of, that would lead to susceptibility of the entire wheat crop because of these dwarfing genes? And you go, well, you know, they've been tested here and there. Well, southern corn leaf blight also had been tested. But the reason that they did not find that susceptibility is because they didn't have the appropriate arrays to test that cytoplasm. Or take sweet corn, for example. The wide use of a single gene, such as shrunken 2 super sweet corn, can we surmise, can we think of the possibility that shrunken 2 could lead to susceptibility to some unknown race or disease? It's possible, right? And so we're saying we learned from our lesson, 1970, and yet we're doing it again. Everybody here loves Honeycrisp, right? How about Honeycrisp? Honeycrisp is now in the pedigree of a lot of apple varieties. Well, isn't that a form of genetic uniformity? And you may say, well, then we shouldn't breed with Honeycrisp because we need to keep the apple crop diverse. But you talk to an apple breeder, you talk to a consumer, and we all love Honeycrisp. It's a productive apple variety. It's got wonderful out of this world tasting quality. Therefore, we will use it in our breeding program. So it goes back to this, be it dwarfing gene, or be it shrunken 2 in sweet corn, or be it Honeycrisp or some other single gene, we use it widely. That leads to genetic uniformity in the same way that Texas T cytoplasm led to genetic uniformity in 1970. And so the point is, one reason that we need to have genetic diversity is to guard, to mitigate, again, for defensive purposes. And having that resource and being able to adapt to unforeseen changes, I think, is a key. And we need to keep in mind that, yes, we need to be aware of these threats, but at the same time, on the other side, there's this pull of increased performance, and increased performance often means genetic uniformity in some form or fashion. So we talked about one use is defensive purposes. Another use is for offense. If you think of a team, an offense, we want higher yield, for example. And I recall the first time that I saw doubled haploids in corn. You spend time in a corn breeding program, and we would have these F1 plants that were uniform, and then you start developing lines. You have the F2, they're all segregating. F3, F4, F5, then they start to be uniform. And the first time I stepped into a doubled haploid field, this was in France, I was astounded. Like, oh my goodness, I've never seen corn this uniform. And the guy I was with, a breeder, he said, yes, that's nice and great, but we're actually concerned about genetic uniformity or the lack of genetic diversity in corn. Why? It's because all these good pedigrees in corn, we're just recycling them to death. We're making process of the same types of inbred lines and the same types of genetic backgrounds. And they're saying, we don't know how long we can sustain this. And if you're a plant breeder in the industry, likely you can sustain it within your career in the industry. So it's somebody else's problem in the future, so to speak. But looking ahead, looking forward, it can become a problem if we keep recycling these lines. Now, on the other hand, if Don Rasmussen were here today, he would say, no, that's not a problem. I've successfully recycled lines from how many? Three, or I don't know how many inbreds there are, Kevin, but not too many. Not too many, just a bunch, and you come up with successful barley varieties. So the idea then is, well, we want it for defense, but we want it for offense, meaning to say that if our pool of genetic diversity for a trait such as yield is decreasing, then we want an infusion of genetic diversity to allow more progress for a trait such as yield or other aspects of productivity. Now, there are other reasons that we might want to use genetic diversity. One is to allow new areas of adaptation. For example, corn became widely adapted in Europe, mainly when they started doing early by late crosses, when they would cross early germplasm from Europe and then late lines from the US, they would cross that, and they found areas in Europe where that type of germplasm did well, and therefore it allowed expansion. So gain in the area of production. Another way, we could use genetic diversity more from a company perspective is to gain market share. We can gain market share, and there could be creative ways to do that. Just this summer, one of my former students, Josh Sleeper, he now works remotely here from the Twin Cities with the University of Florida strawberry breeding program. I go, Josh, that's a great gig because he gets the latest and greatest strawberry materials, and he can grow it in his garden in the guise of, hey, this is an additional testing location. And Josh, I visited him for coffee one day. He went to his strawberry patch and brought to me these white strawberries. And he goes, Rex, these are really good. And he cut up those strawberries and I tasted them. To me, they tasted like pineapples. I go, oh my goodness. I go, this is the best strawberry, I declared, I've ever had. And well, the thing though, is it's white. And so if that ever goes to the market, then that in a way tells of its identity, right? That it's white, and people say, okay, this is that pineapple tasting kind of strawberry. And I think that's an issue though, when we talk about using, genetic diversity provides an opportunity there to differentiate new products that have special qualities from other products that do not have these special qualities. I mentioned Honeycrisp, but I think, the apple breeding world and apple consumers, you're in a good place because when you go to a grocery store, you know the variety you're buying. Is it a Honeycrisp? Is it a Sweet Tango? Is it, heaven forbid, Red Delicious? Whereas in other species, well, yes, the Sparklet pear and the candy grapes are getting there. But for most species, fruits and vegetables, you don't know the variety you're buying. You go to buy some carrots, yeah, it's just carrots. And you don't know what they are. So they could have been bred for special qualities, but that information does not reach the consumer because there's nothing that marks that as being special. And so in a way, if you can use plant genetic resources to have a differentiator for your product that would make it different from the others, the more generic kinds of produce, then that is a way to expand your market share. Okay, so that's the why. A second topic I'd like to talk about is in what packages does genetic diversity come? One such package is known major genes, or QTL. That's one extreme, especially if we're talking about disease resistance, insect resistance, we'd have a known gene, a QTL, a single gene, and that in a way is our package of genetic diversity. Now, on the other extreme would be something like yield, for which there are no, at least for corn, there are no major genes, because any major genes would have been fixed in the domestication process or fixed in the germplasm from continuous breeding. So in the case of something like yield, genetic diversity is not known by a certain gene, any individual QTL, but it's actually known by that line or that inbred or that individual. So it comes in the package of a gene, but it comes in the package of the whole individual, that whole genome carried by that particular individual. Now, there are, however, intermediates between those two, between just major genes and the whole genome. They're intermediates. And one intermediate would be haplotypes. Would there be haplotypes, meaning that it's not one gene, or it could even be a chromosome segment. is there a chromosome segment where anywhere in the genome, in a otherwise not so good line, that actually has value? And the idea then is, well, how do we identify those chromosome segments, individual chromosome segments, and then how do we transfer that to the germplasm that we have? So that's another possible package. And then another package that we have would be an entire chromosome. I've done some work focusing on entire chromosomes, and an entire chromosome is a wonderful package. It's independent from the other packages, so to speak, but it's not a fixed package in the sense that recombination crossing over can occur so that you have new packages at a time. So one way of looking at genetic diversity is looking at individual chromosomes. And a postdoc and I, Andy Thompson, did some work on this. Ines and I are doing some work on this. Basically, you can scan your entire germplasm pool and say, okay, if I look at each of the chromosomes, meaning to say that corn has 10 chromosomes, if I have 200 individuals, that's 200 times 10, that's 2,000 individual packages. Which package might be good for whatever combination of traits I have? And then can we use a chromosome substitution approach to develop germplasm that has this copy of this chromosome from this line now transferred into a recipient line? So that's another package that we can talk about. And finally, there's another package. I've been talking about nuclear inheritance, but there's also cytoplasmic inheritance. And I think the cytoplasmic package, in a way, is something that we haven't looked too closely into. That oftentimes in breeding, something we ignore cytoplasmic effects. And I think for the most part, I think it's probably a safe thing to do, but we could be missing something. I mean, the Texas raised tea is one example, but there hasn't been a lot of literature looking at different cytoplasmic backgrounds and seeing what's the influence of that cytoplasmic background and how might the cytoplasmic background interact with the nuclear genome. So cytoplasm, nucleus, you have individual genes, entire chromosomes, you have haplotypes, you have segments of chromosomes. Basically saying that, well, genetic diversity comes in different packages, different forms, and what form package we utilize would, in a way, depend on what we're trying to achieve. For example, if it's yield, we might not be looking at individual genes, but we might be looking at the germplasm as a whole, or haplotypes, or chromosomes, or segments. Okay, next topic I'd like to talk about is, well, where do we find such germplasm? And if you talk to breeders, I think breeders in their minds, at least in the corn world, have a general ranking of the usefulness of different types of germplasm. And for a corn breeder, at least during my time in the corn seed industry, and it was allowed at that time, a most useful source of genetic diversity was competitors' germplasm, competitors' hybrids. And so when I was with the seed industry, it was a fairly common practice at that time that you would take the latest, say, Pioneer hybrid, and you would self-pollinate that Pioneer hybrid in the hope of getting something good and useful. And guess what? Yes, that you can get something good and useful even from selfing a commercial hybrid, which you know is mixing the heterotic pools, but still somehow you get something. You can get something good and useful. In fact, we had a joke that, well, that is a successful source of germplasm that even the pioneer breeders were doing it, that they were selfing their own commercial hybrids. Of course, that wasn't true, but we were kind of tongue-in-cheek joking that that was what's going on. And sometimes in that world, we had to, because we didn't know about, well, what's the genetic background of this germplasm, we were thinking of creative ways to say, well, how can we look into, the genetic background of this hybrid? And one idea that we tossed is, the pericarp is maternal in origin. What if we analyze the DNA of that pericarp? And then we analyze the DNA of the plant as a whole. And by subtraction, we know the female parent, the pericarp, and by subtraction, we can know the male parent. So, when you're in that situation, you think of roundabout ways to accomplish what you want to do. Now, In this day and age, though, I think with many, at least in the corn, many of the lines being protected by a utility patent, then you can't do that anymore. Which brings me to a bit of a segue is when working with germplasm, just the legal aspects are something that are extremely important. I'm speaking from experience here because in my hand here is some germplasm and some North Korea. So North Korea, this is Korean high oil. It says it has 20.91% oil. And this is regular corn belt dead corn has 3.5% oil. So 21%, 3.5%. And you may say, how did we get this North Korean high oil germplasm? The story is that time we had an adjunct faculty member in our department who was doing, she's from South Korea and doing agricultural development work in North Korea. And so she was an intermediary, and she got from the North Koreans this germplasm. They apparently were selecting for larger embryos. The oils in the embryo select for larger embryos to increase the oil. And the idea then was to give this seeds to us so that we can do some work on it, and in a way that would somehow will funnel back the benefits to the North Koreans. There was no paperwork, no materials transfer agreement. It was, I think, just one USDA APHIS type form. And so when we talked to our Office of Technology Commercialization, they said, no, we're not going to touch that. We can't touch that because of the lack of information on the freedom to operate. Then I talked to somebody from the USDA, a curator of the maize germplasm collection, and I told her about this. And she said, she's retired now, oh, just give the seeds to us and we'll make it available to anybody. I go, I don't know if I should do that. And so I was thinking, well, maybe I should just wait for institutional memory to fade and wait till people forget where this came from. And they go, oh, by the way, I have this germplasm. And by now, the institutional memory has faded. But then again, well, then what do we do with this? Has it commercial value? Not sure, because there's high oil out there. But the lesson is we're stymied in our ability to work with this germplasm because of the lack of paper development. So if you remember any one thing from today, make sure you do the paperwork. Make sure you do the paperwork. It takes time. I just learned this. I requested some seeds from Tanzania, and boy, that was a very interesting and learning process to get the seeds from there to finally here by FedEx in Minnesota. And for good reason. We want to protect the nation from pests and pathogens, but make sure you do the paperwork. Talking about paperwork, another source of germplasm would be of genetic variation would be germplasm whose plant variety protection certificates have expired or whose plant patents have expired. So plant patents are for asexually propagated species. PVP are not for the sexually propagated species traditionally, but now they include asexually propagated species. And so what I heard is in corn, it's become a very valuable source of germplasm. Yes, the germplasm could be old, about 20 years old, but I've heard that their companies are actually on their calendar already. They put on the calendar the date that this that protection expires, so that they can go to where they need to go to get seeds right away, right after that expires. So that has proven to be an effective source of germplasm. Another source of germplasm is germplasm that is elite somewhere, but not where you are. And that has been found useful in corn. For example, corn breeders in the US, they found that germplasm that is elite, for example, in Latin America, that's been valuable in many ways to the corn germplasm. Now, there might be some challenges with that, but at least, it's elite somewhere. So what it would take is, adapting it to the US, incorporating it to our germplasm here, and then good typically comes up. Now, going down that ladder, so to speak, a next source would be the older germplasm. The older germplasm that you typically don't use anymore, the germplasm in seed banks and germplasm collections, that would be a next possible resource. It's funny how oftentimes we call them a seed banks, but in many breeders, they call them a seed morgues, in the sense that, yeah, the seed goes there, but it never comes out anymore, and it basically dies there. And so the idea then is, well, can we make that less of a seed morgue, and can that be a seed bank, and what are the effective ways we do so? So I've talked about these sources of genetic diversity more from the whole germplasm, the whole genome, from individual level. Now, if we look at the other extreme, then there could be sources of genetic diversity from gene editing. We all know that. And of course, you need genomic information on gene editing. You could do traditional mutation breeding as a new source of genetic variation. We tried to do that in the community plant breeding team, the vegetable breeding work that we're doing. Before we were successful in getting germplasm from Africa, and our germplasm base was narrow. So, well, why don't we just nuke this stuff and create new variation that way? And so there's a facility in UC Davis that actually does that for a fee. So we sent the seeds there, but not having developed any protocols, we used their default protocol. And when we got the seeds back, guess what? 90% of the seeds germinated. And we go, that's too high. That's too high, and we should have been more intense in the treatment with radiation. But for a variety of reasons, I don't think we can go back to that source. Well, I'll tell you why. We had an issue with the payment of the bill, and in the other end, they didn't charge us, and we didn't pay the bill, and it kind of iffy to, hey, we did pay that bill, but can you do this work again for us? So I don't think we're gonna. All right. Now, I think the last part is how do we use this genetic diversity? If you're talking about individual genes, if you're talking about QTL, then it's pretty straightforward, right? If either the gene is known, or you find germplasm that would have, say, the resistance you're looking for, you do a mapping project, you find QTL, and then you transfer it, say, by backcrossing to your elite germplasm. Once that gene or QTL is now prevalent in your germplasm, you might not need to do that anymore, because the new parents you use would carry that gene. So you would make crosses like you normally do, and just select for that gene as it's segregating. And then if both parents have that gene, then you don't even have to worry about it anymore. It's naturally carried in the germplasm. The greater challenge, I believe, is more for quantitative traits. For quantitative traits, for yield, for example, well, how do we effectively transfer genes from an unadapted source that is not rich in these good alleles, but has something? How do we do that effectively? And so just to illustrate why that is difficult, let's say that I have 100 QTL 1, 2, 3, up to 50, and then 51 all the way to 100. And let's say that this is parent 1 and parent 2, and parent 1 carries the allele, the positive allele at 1 to 50, okay? And then the minus or the less favorable allele at 1 to 100, at 51 to 100. And let's say parent 2 now has negative for the first 50, and it carries the positive allele at the remaining loci. Meaning to say that the two parents complement each other, and they carry an equal number of favorable alleles. So if you make this cross, 100 QTL, let's just say that they have largely equal effects, and then you ask the question, well, if you plot the number of favorable alleles in the population, what would the mean be? Every 50, right? Probability of 50 in one locus. You get homozygous lines. I'm sorry, these are homozygous lines. On average, a line would carry 50, would be fixed for the plus allele at 50 loci. It won't always be the same out of the 100, but 50 of them. Any guess on what the upper limit here is? Huh? Oh, you can't get 100. Well, you take a really, really, really large population. This is about 70, 75. Let's just say 70. 70%. Meaning to say that you can get an individual fixed for the favorable allele of maybe 70%, 70 of 100. And here that would be 30, okay? Now, in this case, there is a high probability of genetic improvement. Why? Because we get genetic improvement if we get something better than either parent. Meaning to say that if we get a line with 51 or 52 or 53 alleles that are fixed, we have genetic improvement. In other words, this is the area for genetic improvement, right? So yeah, that's half of the curve. So we cross good by good, chances of getting something better are actually quite high. Now, let's say that we're now working not with two equally good parents, but with germplasm that is one's a lot better and the other's worse, but still has good alleles because it's, say, exotic germplasm. So let's say that this is now 69, 70, okay? And now this is 71 to 100, okay? So the first parent is fixed for the favorable allele, 7 here, and 71, okay? Fixed for 70, and then the minus allele at 30, and then the poorer parent is minus at 70 and fixed for 30. In this case, if you make this cross, then the mean remains the same. The mean remains the same. But we have genetic improvement only when we get 71 or more. Okay. Because we need to be better than the better parent, we get genetic improvement if we have 71 or more, meaning that the space for genetic improvement becomes much less. And that is why we like crossing good by good. And that is why it is so difficult to incorporate exotic germplasm from a poorer source, because as we do so, well, yes, we're losing the alleles that we already have, the good alleles that we already have. And therein lies the challenge, I think, in utilizing exotic germplasm. Still is an issue today. You know, how do we do that effectively? But in closing, I guess my way of thinking is we now have a lot of technology at our disposal today. We have a lot of technology. Are there ways to mitigate this? Are there ways to effectively transfer the good alleles from a poorer source while keeping the good alleles that we already have? Possible ways to do that would be being able to control recombination. And so, you know, can you do that? CRISPR can do that to some extent, but are there other ways to do that? Can you use, say, machine learning or AI so that at each generation it would tell you, okay, your probability of success will be better if you keep this plant and you cross this plant with this other plant, you might get something, the best combination in the end. And so to me, I think this is a very fertile field, because with this area, this time we are in which things like climate change that are a big concern, and producing more food for a growing population with less resources, how do we do that effectively? And yes, we can continue to do that just by continuing to cross good by good. But I think in the long run, it would benefit us a lot if we have a broader germplasm pool. But to do that effectively, then we need to be creative. We need good, creative ways to be able to effectively harness the genetic diversity we have that's there, so that we pick what we need and keep what we already have. All right. That's all I wanted to share this morning. Thanks for being a receptive audience and for bearing with me without my, well, not only one slide. Well, thanks so much. No questions.
Was that a statement or a question?
I don't know.
I have a question.
Yeah.
So do you think like we should improve more in the parental selection, because like if you go back, for example, we do genome selection in F5, for example, we have 3,000 lines, but then 400 lines, for example. So all those extra lines, we already have genotype data on them, but we'd be able like to improve, use what we have to predict offsprings, because for example, you mentioned the honeycrisp. So you always want to keep finding the best lines, but best line not always deal with the best offspring. So what if we can find a way to like predict the offsprings, so we can keep the genetic diversity within our breeding lines, but with the resources we already have, don't need to keep adding.
Yeah, I think two things. Prediction works best if you have strong relatedness, right? And so meaning to say that if you're relying on predictions in your breeding program and you want the predictions to work, it's better if you focus on related material. And so, you know, in terms of expanding genetic diversity, then it's possible that you consciously or unconsciously would be decreasing your genetic diversity because you want predictions to work and you're using germplasm in which those predictions work best, and that would be the related germplasm. Second is, well, how do we quantify, you know, or it's not how, but when we select for performance, do we also select for diversity? And I'd say that typically, no, we don't do that. I'm aware of some breeding programs that they've started to do that. They've started to tell their breeders, okay, when you make the breeding process, yes, pick the ones that would give you the best mean performance, but we should also have metrics to measure the diversity in the crosses you make, so that we can track over time, you know, are we losing more diversity than we want to?
Given the current technology, what do you think is the best way to identify and utilize favorable germplasm in a diverse background for an quantitative?
Sounds like a prelim question. I don't think it's I don't think it's just any one technology, but a combination. So you would need to start with good phenotyping, good characterization of the germplasm you have. I think it starts there. Obviously, you'd need genomic technologies to be able to dissect the diversity that you have. And I think it's not really identifying, that's still a challenge. For me, it's more of once that diversity has been identified, how do we incorporate that effect? Because, you know, as you and I know, well, yeah, I could have for my entire genome, I could say chromosome 1, you know, these are the positive alleles marker-wise, and these are the negative alleles. But then how do I just use or capitalize on the positive ones without the negative ones? And there have been some proposals that have been put forth. For example, Alain Jacques-José, who was a visiting scientist here, he and his group have been working on this concept of bridging process, so to speak, that you have your elite germplasm here and you have your less elite germplasm here. Is there germplasm that actually is an effective way of bridging that gap? Kind of like in, say, the Tour de France. If this is the peloton or you get left behind and this is the leaders, typically you can't just make it to that. Somebody has to go back from that leading group and then it drag you back to that. So it's kind of the same way that oftentimes maybe you need that bridge. And if so, what would be characteristics of good bridging germplasm? I don't know what that would be. But anecdotal evidence seems to indicate that there's germplasm that performs better than others for that bridging that I just described.
Got a couple of online questions here. Could you speak to managing diversity versus trait improvement in species that are largely outbreeding and do poorly as inbreeding increases?
And I speak on genetic diversity versus trait improvement in outcrossing species.
Yeah.
Okay. Pick another crop.
Relatable.
That's the first thing that comes to mind, but no, I'm being facetious. Well, I think it's more, in that case, your challenge is that, I talked about how genetic diversity comes in packages. And oftentimes it comes in, if it's a self-pollinated species, then a package would be an individual line, for example. And in this case, then if you have an outcrossing species, then that package would be the population as a whole, and then the individual lines in that population as a whole. So you have, and then knowing that that individual cannot be replicated unless you can asexually propagate it. So you're dealing with another level of complexity or logistical issue anyway, in that, yeah, you don't have as nice a package of genetic diversity. And so you need to maintain large population sizes for one thing. And but number two is then within that large population size, you could have opportunity for selection. And you'll need to think, okay, do I gain more by selecting within a given population or between populations, or what ideal combination of selection do I use between and within the population?
Do you think that IP agreements and MTAs will inevitably lead breeding programs to a bottleneck?
Well, we already have bottlenecks, I think. IP agreements are in place. That's a given. And we already have bottlenecks, and IP only serves to make getting out of those bottlenecks harder, I think. Just because, again, of freedom to operate or not being able to access germplasm that you otherwise might want to.
I had a related question. I had a question related to that. So do you think, I mean, barriers for team germplasm because of IP, think it's a net positive or a net negative? Maybe, but clearly it's a net positive in the private sector. The public sector is increasingly difficult to share germplasm too. Do you have a feeling or opinion on whether that's net negative or net positive?
In the private sector, yeah, I agree. It's a net positive because a strong IP in place has allowed investment in the breeding programs while by being able to be in a position to recoup those investments. And I'm aware of situations in which companies had to shut down breeding programs in countries where IP protections were not strong, and they were forced to just shut it down because they can't recoup their investment. In the public sector, I don't know how, maybe you're more familiar with this or others are. I don't know, are IP agreements that difficult? I mean, they're a hassle, right? They're a hassle, but I don't think they're overly, once you've done it a few times, and once you've had that relationship in place, I think maybe, your first IP agreement with another organization might be the toughest one, but once you have that established, I would expect that succeeding IP agreements would be easier to navigate than perhaps the first one. Yes.
Do you think that you spoke about bridging germplasm? What about bridging environments?
It's a great point. Yes, bridging environments. That's another key. I think is that with things like maturity, photoperiod, and sensitivity, that yes, use of bridge environments could be helpful. Either natural environments or controlled environments. And that's a wonderful point. Yeah.
I'll ask a related question that we've been going through. So related to these bridging environments, if you were to test photoperiod sensitive materials there and consider converting them to photoperiod insensitive, to what extent do you think we can use genomics and phenotyping to predict which lines will perform best once they're converted?
Well, depends for predictions, depends how well photoperiod or not, depends how well that germplasm is represented in your training population. So if it's new germplasm, it probably, it won't work. And so the first boils down to, effective phenotyping to start with. And of course, if you have issues with photoperiod, then the phenotyping will not be meaningful until you take care of that problem. Then phenotyping can become meaningful, then predictions can work after you accumulate the phenotypic.
So So the students and postdocs here, they'll still be breeders in 2060, I suspect, 2050.
Okay.
And I'm betting that by 2050, we'll be able to genome edit most of the genome, or just synthesize it from scratch, one or the other. And there'll be like some serious databases that'll say, here's 30,000 genes, this is what they do. And then there's RNA genes, here's the list of what they do. So will we still have breeding, or will people just sort of like say, well, I'll take these, of those 100 genes, I'll just sit and fix those 30, and then we're good to go?
Yeah.
How will that change things?
Well,
What genetic
no,
resources are?
I think it's a great point. I thought of genome editing, for let's say for yield, and let's say you can edit the entire genome. I thought of that issue as something like analogous to a really advanced sound mixing, where you're looking at the individual frequencies and you're cutting out frequencies and you're boosting some. And if I have 30,000 or 10,000 of those frequencies, that would take a long time to optimize each one of those.
But I only need 30 for that.
Well, if it's only 30, that's easy. But if it's a large number, then it becomes harder. So that's one. But to your point about breeding, and so this goes a bit more. It's something I've wondered about. How many breeders will we need in the future after this generation here? Okay, with technologies, for example, in AI, can a machine replicate a breeder's decisions? I think it can. Not perfectly, but can it replicate, well, 80% of the decisions, so that the machine can do the initial cut, and a fewer group of breeders then can look at this reduced number of lines and make their choices. I think there are possibilities there. And if you think about it, well, you don't have to pay fringe benefits or health insurance on machine. And so I know it might not paint the most rosy picture from a human resources standpoint, but I think it's a reality that's worth thinking about. Which also brings to mind, well, and I ask this from an distributor. So, if you have all these technologies, all AI and machine learning, what's a breeder these days? And he chuckled and he go, I wondered the same thing. So, because some of the tasks that traditionally, talk about corn here, a corn breeder would do, well, many of them are automated now. And of course, you need human intelligence and expertise to continue to fine tune whatever's automated. But yes, a lot of it is automated. It's a great question. I'll be retired by then.
Another question. So if we have time, clearly a lot of contacts in the industry, a lot of insights into the industry. So, and for a long time, people have been talking about the narrow genetic base of corn, so this industry. Fields keep going up.
Yeah.
And new products being developed. So you think there's going to be a reckoning that you'll have to think more seriously about diversity, or are they now, or what's the current take on it?
For corn, I think it'll take a long time. Corn is just, even though there are a few lines, even though there are only a handful of lines that are pedigrees that are important, maybe 10, a dozen, 15 or so, they are diverse already. So there's a lot of diversity there. And then my mind goes back to the Illinois breeding long-term selection program for oil and protein. You have a single population, first white, that has been selected for many, many generations for oil, for protein, high oil, low oil, reverse high oil, switch back high oil. They do all these sorts of different things. And yes, there's a lower limit of close to 0% oil or protein. The machine can't measure it well anymore, or the plant just needs a bit of oil and protein to live. From the high direction, the gains continue to go on. And if you think of, well, if that's oil, there probably are more genes for the yield. And I think we can continue to keep going actually with the germplasm base, if that's what we decide. But then the question is, well, can we get even more gain if we diversify the germplasm is. And I think the answer is yes, except there's a lot of catching up to go, a lot of catching up to happen that needs to happen because of the head start of the current germplasm.
Go with that one.
Okay.
Oh, yeah, okay, I've got a couple related to parent selection on mine here. While elite by elite crosses work, the available elite genetic variation is limited by recombination events, which are biased based on combining ability. From a practical sense, how do you envision breeding programs identifying parents that tend to generate more genetic diversity or variance?
Oh,
It's a tough question.
I don't know how to answer that one. I mean, you could certainly look at the predicted mean performance. You could look at the marker diversity between the two parents. Yeah, I think those are the basic tools that just by maintaining, looking at the diversity between the parents. But not only that, because then you're looking at just a very small slice of the whole picture. You need to look at the diversity of your germplasm as a whole. So I think what might need to happen is just in the way that breeders keep track of the annual mean gains they have in productivity, it'll be important to keep track of the diversity that is maintained in that germplasm from era to era, from decade to decade, from year to year. Hey, that sounds like a wonderful thesis project, right? Yeah, I should go back historically and genotype germplasm and see the gains and see, hey, how much diversity have we lost or gained as the years have gone by?
Need more?
No, you could take
I
questions.
just have kind of a silly question.
Hey, Bob.
Because you brought it up. You brought up Korean high oil.
No.
And you brought up long-term selection trials, Urbana.
Yeah.
Here's my question. I heard a rumor about 15 years ago. Some people believe that the Korean high oil may have been derived from that long-term selection experiment. You heard that rumor? Any truth to it? What do you think?
So yes, we heard that rumor. Yes, we tested that hypothesis, different origins. We analyzed, we looked at molecular marker data. And of course, that was white germplasm. This is yellow, which some indication, but not complete indication, could easily change color. But yes, we did look, and that doesn't seem to be the case. If you want some seeds to further, this can go within the university.
Do you know if that corn also has high vitamin E levels?
Ooh. You want to join the University of Minnesota? No, I don't.
Yeah.
Can you look at? Let's talk. Can you analyze that with ground material?
Yes.
Oh, okay. That's where I go. Other questions? Cheryl.
When you think about implementing like a two-part breeding program where you have different selections, make lots of crosses one part and like internal development on the other part, do you think it would improve the chance of getting genotypes with lots of beneficial items?
I think there's value in that. So the idea is that part of the breeding program is like continuous population improvement. And the second part is development of finished varieties from that population. It's no secret that we've been talking about it. Bayer is, in a way, Bayer is organized like that right now. And here's their thinking, and I actually think it's brilliant. What they're saying right now, how many breeders think is, well, what I select in my mind should be potential varieties in the future. That would be my criteria for selecting potential varieties. In that thinking, it's not, you select not based on potential varieties, but on potential, its potential as a parent. Meaning to say, yeah, it could be really good for 3 traits, maybe not so good for 1 trait. It would never make it as a variety, but as a parent, it's actually quite good. So you select that and you include that then in that population. I think that's a good way to proceed. Again, just in our mind, separating parents versus finished varieties. Okay, well, time is up.
Thank you, Rex.